The next topic, the next technique is tissue culture. Tissue culture is the growth of cell or tissues in an artificial medium under sterile condition. Artificial medium mean that is the nutrient medium which contain all the macronutrients and micronutrients that are necessary for the growth of the plant. Thus, this nutrient medium may be solid, may be semi-solid, or may be liquid. For example, agar can be used as a nutrient medium. Broth can also be used as a nutrient medium. Sterile condition mean that the atmosphere or the area or the place where you placed your tissue culture plate or tissue culture container, that must be free of any contamination. Tissue culture technique can be applied both for plants and animals. In case of plants, it is called plant cloning or micropropagation. The reason is that from a tiny cell, we can generate thousands of identical plants. These plants, this is one of the best method by which we can develop thousands of disease-free plants from a diseased plant. This diagram shows us some of the important steps followed in the tissue culturing of plants. This is a sample, the tissue sample scraped from the parent plant. This sample or scrap tissues are grown in the nutrient medium, that is agar, plus oxygen is also applied. Oxygen is the growth hormone. After some time, the tiny plantlets grow. You can see here. These tiny plantlets are then further transferred to the compost. Compost is usually the nutrient conditioner for the growth of the plants, or you can say that it is the conditioner for the soil. Tissue culture can also be employed in animals case, but in case of animal we cannot call it as animal cloning, because in case of animal we simply from a single cell we can make a layer of cells on the container. The tissue culture in case of animal, we use it to see any type of abnormality in the tissue or in the cell. For example, widely used in cancer or chromosomal disorder. From the organ, we can isolate a cell and then culture it. After culturing it, we subculture it, and further we obtain a cell line. So by the help of tissue culture technique, we can get our desired plants or desired cells from the in case of animal tissue culture and in case of plant tissue culture as well.